Okay, so here we are, and we're going to talk about the Sentinels of the lab and ensuring safety across the galaxy. So why am I always talking to you about safety and bioterrorism kinds of topics? Well, there's a couple of reasons. Just in case you don't know, part of my funding for my job comes from the Public Health Emergency Preparedness Program, and we are held to a couple of different basically goals that they set to us that we have to do, and that involves training and talking about biosafety training, and we have to report how many we do and all of those good things so that we show that we're actually using the money appropriately. But another reason is that I've just been really fortunate in the time that I've been at the Public Health Laboratory. We've had, obviously, a lot of pandemics and just different outbreaks and things that we've had to deal with in that time. And it was during the Ebola outbreak or pandemic that they were really concerned about biosafety. And so they put a lot of money out there. And at that time, I was fortunate to get a lot of training, and in turn tried to provide that training to all of you. So we've done things like working on risk assessment and other things like that. One of the things that keeps coming up, though, recently from laboratories, and I know this isn't going to affect all of you, and for those of you who don't even work in a laboratory, this talk still is really important to hear so that you understand better what happens when laboratories that are doing full microbiology potentially start dealing with a possible bioterrorism agent in their laboratory. And so these laboratories were asking for a little help because they don't see a lot of them, but when they do, it's pretty stressful if they think that there may be exposures in their environment. And so we're going to go ahead and work through a little bit of biosafety today, and then working with these suspect BT agents and exposure assessment. So here you have our Princess Leia talking to all the troops, trying to share their strategic plan. And a strategic plan is part of any initiative. That she wants to keep, her rebel forces safe against, and all the Jedi safe against the dark side of the Force. And in the laboratory, we put together something that we call a biosafety plan that's supposed to help all of us stay safe. One of my favorite things that I get to do in this job is when I get to actually get out into laboratories and visit you in your facilities and talk to you about biosafety. And one of the questions we ask, we have a little survey that we complete, and I'll ask, do you have a biosafety plan in the laboratory? And often I will hear from people, oh yeah, we have this hospital plan, and people start talking about their hospital safety plan, which is great because it has a lot of information in that the laboratory needs to know. But what isn't typically in that plan is information that's specifically relevant to the laboratory. It doesn't talk necessarily about keeping laboratory staff safe. It's about keeping your patients safe in the hospital and the hospital plan. So you really need a biosafety plan that is about laboratory and how you're going to keep functioning in the laboratory and how you keep your own personnel and staff safe when there are things going on. So then I'll start talking about, well, what does a laboratory biosafety plan include? And it's like pieces of a puzzle. People will start saying, oh yeah, I have this, but it isn't really pulled together in one place. And so it's like the puzzle pieces, but the puzzle hasn't been assembled yet. And what I really want to impress on this is that if you don't know where your plan is in your laboratory, or if you even have a plan, go back and ask your supervisor or manager, do we have a specific laboratory biosafety plan, and where would I find that? So in my perfect biosafety plan for the Jedi warriors and for you in the laboratories, these are some of the components that I would like to have you put into your biosafety plan so that they are all there. And the first one would be vaccinations. So most of you give some kind of vaccination to staff. We know hepatitis B is out there. Often we may have meningococcal types of vaccines. In our lab, we also have rabies vaccines and some other things. So whatever those vaccines are that you offer staff, where do you keep those records? Maybe they're employee health, but just the fact that if you would have a listing of what's offered to staff so that they know what keeps them safe, that would be a great thing. Biosafety training. This is something that is really encompassing of a lot of different things, and I don't really have time to go into all of the different components that would be in there. But we do know that even on the first day of work, there are certain things that you have to go over with a new employee about biosafety and working safely in the laboratory. How did they do that? Where are the policies? What PPE do they need to be wearing? How do they don that properly? How do they doff it properly? Just how do they work in a biosafety cabinet safely? Do they do it correctly? So that happens with a new hire, and then annually we all have some type of refresher training on biosafety. And so we have this training piece, and what is in there, that would be part of the biosafety plan. We talked a little bit about risk assessments, or I mentioned it at least, that all of the work that we do in the laboratory, it's become important that we realize now we need to look at it step by step and identify any hazards associated with the work we're doing. Often for a biosafety level, we're looking for things like where can you get punctured with something, where are you going to potentially breathe an aerosol, those types of activities that you would be potentially getting exposed to an agent, a biological agent. But whatever that is, that risk assessment, then once you've identified all of those hazards, you go ahead and you look at the likelihood of them happening and the consequences. How consequential is this? Is this something somebody's just going to get sick? Is this something that they could get seriously ill? Would they be hospitalized? You will come up with that, and then you're going to go back and you're going to put into place the next piece, the mitigation measures that you're going to put in place in your laboratory to prevent those risks from actually happening in your laboratory. So you do this cycle, and for most of us, we didn't go back necessarily and do all of a risk assessment on everything we're doing, but hopefully we're doing it on new tests as we bring new tests into the laboratory now, that risk assessment piece is just part of the actual validation and verification of testing that is there, and that's a piece that's there. So we should have those risk assessments, and it isn't enough to do it once. You always have to go back and reassess those risk assessments, look at them again. Has anything changed? Have you moved equipment? Do you have new personnel? Whatever that is, you need to look at those things and check them out. Then another piece, though, what I really want to highlight in today's talk is the information that I have in the bolded print here. So contact information of who are your partners, who's your occupational health team, who do people call when something happens, when they suspect that there's been an exposure incident. And they need to know who the people are and how to reach them, and not just on first shift, because these accidents happen on weekends and second shift and third shift. And I can go into some stories later about where that hurts when people don't know where to go. But if you can have that all in one place and people know where that is, that's a really valuable piece of information in that biosafety plan for your staff. Incident reporting goes hand in hand with that. When something happens, there are typically forms in every facility that have to be completed and turned in within a certain amount of time. Do staff know where those forms are, what they have to do with that, and what kinds of things are they reporting? And then actually assessing the exposure and monitoring exposures if they've happened. Next, we'll go to a root cause analysis. When there's been an incident, you want to go back and look at why it happened, and not just ask why once, but several times. And again, having those partners doing that outreach ahead of time so that all the partners in your occupational health plan know who they are, what their role is going to be. So that's all done ahead of time. And then potentially after action reports, and then making sure that all employees know where this biosafety plan is kept. So let's go ahead and start our Jedi training. We'll have Yoda here leading us into how we can stay safe. So we'll start out with some iClicker questions. And the first one is, what sources are you most likely to recover a select agent from? Is it A, blood culture, sterile body fluids, upper respiratory specimens, B, lower respiratory specimens, sterile body fluids, tissues, C, lower respiratory specimens, wounds, blood culture, D, wounds, sterile body fluids, urine, or E, blood culture, wounds, sterile body fluids. Okay, still a few of you to vote yet here. Okay, let's see. We'll stop the voting and, okay, the majority seem to say C, but we do have answers all across the board, so we're not necessarily real certain of this answer. And the actual answer is C, so good for those of you who chose C. It would be a lower respiratory specimen, wounds, and blood cultures. So you need to talk to staff about this, again, as they're training and learning how to read plates and things. Some of the key sources that they want to stop and ask, if this is a potential bioterrorism agent, that would be looking at that lower respiratory, whether it's tissues or fluids from that area. It would be the blood cultures, and possibly it could be sterile fluids that you inoculate into your blood culture bottles. And then we know that wounds are a possibility. We just heard Beth talk about the cat bite, so we've got our lovely little cat there to remind us. Lots of cat pictures. Okay, next question here. You are reading plates, and you have a joint fluid culture with no growth on blood or McConkey's and slight growth on chocolate. What do you do? So A, do you run it on MALDI? B, do you gram stain it and then run it on MALDI? C, you run it on MALDI and then do a gram stain. D, you close the plate, check the age of the culture, check the patient history, gram stain, and then run it on MALDI. Or E, do you close the plate, check the age of the culture, check the patient history, move into a biosafety cabinet to do the gram stain, and dependent on the gram stain result, stop and perform rule-out testing? So this one should be pretty easy, everybody. I think I've been pretty clear. Okay, so let's see. Yay, the majority got that it would be E that I am looking for on this question. So you want to always close the plate quickly. You want to check, go into your chart and check how old is this culture anyway? Why isn't it growing well? Can you get at the patient history easily or not? And then just move into the biosafety cabinet and do any work you're going to do in there first. So do your gram stain. And then if you're seeing something that looks suspicious on that gram stain, stop. And don't put it on the MALDI. Please don't put it on the MALDI. And then perform your rule-out testing. So these are a couple of clues that you could be working with the BT agent. And you have a blood culture that's taking longer than 24 hours to grow. There isn't really good growth, or it's just a slight haze of growth at 24 hours. If you're seeing better growth on chocolate than blood, if you have a gram stain that's showing tiny gram variable coccobacilli, if you have a gram stain that shows large boxcar shaped gram positive bacilli, potentially with spores, and then checking the patient history, going in when you see those clues, and can you check, do you see anything in the notes that says that the person has traveled to an area or has lived in a country where BT agents are endemic, or that the patient has had some kind of an insect or an animal bite, or just that they work with animals. There is a really nice, so this is one of the first times we're going to go into this folder here. Again, this is something that many of you have asked me to look at and address, and so at a national level, we have worked to put together this little stopping point thing that actually goes through a lot of when you need to work in a biosafety cabinet. So let's see, I think the slide is up next. And you can't really see it, but you can look at it here in your folder. So it starts with working with the blood cultures and thinking about, okay, how long did it take for that bottle to turn positive? How long was it incubating? All of your normal pathogens are going to turn positive before 24 hours. If it's something that's taking 24 hours or longer, right off the bat, that's kind of a flag that you may have something you need to worry about a little bit more. And then just working in your biosafety cabinet, what are you seeing on those gram stains, which I mentioned? And that flows into the subculture plates and where you actually have plates and on cultures. So it all flows together. Hopefully you will find this to be just a helpful little job aid that maybe can be posted or gone over with staff as they start working up cultures. And this job aid is actually going to be included as we're revising the, there's a preparedness guidance, emergency preparedness guidance document that we put out several years ago that we're working on revising right now. So this will be additional in that when it gets released, hopefully early next year. So, okay, so this is just my reminder slide to not use the MALDI until you've ruled out BT agents. So often when laboratories have exposure, it's because we understand that you are concerned about patient care, and the fastest result that you can provide to the physician is the best that you feel like you can do to help that patient. But you need to know when to just take that step back and slow down. Also, the MALDI isn't very good at identifying these bioterrorism type agents. It will either call something a bioterrorism agent when it isn't one, or it'll call it something similar, and you may not even know you've been working with the bioterrorism agent if you don't catch it. So it's just not a good idea to use the MALDI if you have any questions at all. Another place where laboratories often report to us that they get caught are in doing these rapid blood culture identification systems. So they've got a positive blood culture bottle, and the fastest way to get the doctor a result then again is to do something like setting up the Verigene or a BioFire panel directly on that blood culture bottle instead of waiting for the culture to grow the next day. But if you have pieces of that you're performing outside of the biosafety cabinet, then potentially you also have aerosols being generated as you're setting up those tests. And so if you can do any of that work in a biosafety cabinet, that would be advantageous to you and prevent some of those exposures. Also remember, if you're a laboratory that is just doing the initial reading of a positive blood culture bottle and maybe subculture, and then forwarding that on to another laboratory within your system to do that full identification. Please, please, if you do see anything that you suspect at all could be a bioterrorism agent from that gram stain, if you're having trouble with that gram stain and you know that it's a slow grower, give them the heads up on those plates. We've talked about this in the past, but it really can prevent secondary exposures at another laboratory where if they don't have any idea that this is coming, they may have those plates on an open bench that they're reading them. And just talking about rule-out testing really briefly, when you're doing your rule-out testing, you always want to work in a biosafety cabinet, and if possible, use biosafety level 3 biosafety practices, which would be including some kind of a set of safety eyewear, a face shield or something, the N95 mask or a PAPR, and then to add the additional gown that would be opening in the back, and your gloves. So those are things are kind of standard. And the minimal testing that we really hope that you can all perform is the catalase and an oxidase. And catalase, if you're going to do the catalase, I know most of us are more familiar with just doing a catalase on a slide out on the open bench and just putting a dot down and adding some reagent to it. But the safest way to do it is to do it in a tube, actually, where you put the hydrogen peroxide down into the tube, a little bit of it, and then you just put the organism on the side of the tube right above where you have that reagent, and then just tip that tube up and watch for the bubbling that would occur when it hits the agent. So either way is acceptable. It's just a less risk of an exposure when you do the catalase in the tube, so less aerosol production. And then additional testing, rule-out testing, that's really helpful if you can keep these in your laboratory would be motility, and again, a tube method would be safer than to do hanging drop motility or something like that. And then having urea, indole, beta-lactamase, and the ability to do some satellite testing. That gets you pretty well through the flow charts that you're going to need to do your rule-out testing. So let's go to our next question. What would you do if you realized you were working outside of containment with a possible BT agent and had been exposed? Would you A, call for help from the Rebel Alliance? Would you B, ask your coworker what to do? Would you C, notify a supervisor and ask for help? D, would you go to your biosafety plan and turn to the laboratory contact section to find the list of who to contact and how to reach them? Or E, would you go to the emergency department at the end of your shift? Okay, so let's see what the group said. So for most of you, you chose the D, going to the biosafety plan and turning to the laboratory contact section to find the list of who to contact and how to reach them. And that's really great. If you have a plan like that, you may not have that kind of a plan. So if you don't have it, that's something to think about taking back and getting set up in your laboratory so you can do that. Oops, wrong way. Okay, so who is on that team of partners? Beth already did a nice job of laying that out from her case study. So you're going to have you as the clinical laboratory, you're going to have us at the state public health lab, you're going to have people at the state epidemiology and the state vet, as I said. You may have your infection preventionist, anybody else on your occupational health team, your employee health, a whole bunch of people. And it's just really important that they know who they all are and what's going to be expected of them when something happens. It's also really helpful if there's an event going on that everybody can see the big picture and understands what's going on and can have a chance to ask questions. So think about how you're going to communicate. Are you going to have some type of a meeting in person? Are you going to do a Zoom type of a call? Anything that you can set up just to offer people that opportunity to ask the questions, and then maybe provide some guidance if they're not sure what should be happening next. So this is just a wonderful way to meet and handle these situations. So in the event that an incident occurs, do you know what to report and what forms you need to complete and where they are kept? So A, yes, you know what to report and the forms that you need to complete and where they're kept. B, yes, I know where the forms are. I know there are forms I need to complete, but I don't know where they're kept, and I'm not sure exactly what I need to report. C, I don't know anything about reporting or what forms I need to complete or even where to look for them. And D, that information is classified. E, my supervisor will take care of this, and I don't need to know. So which is your choice? Okay, So the vast majority of you know that there are forms where they're kept and that you need to complete them. Some of you know that there are forms and not quite sure where they're kept, but I like that we've got the across the board answers and that there are some people that think the information is classified. So incident reporting, this is another interesting thing that they talk a lot about when they're training us as biosafety officers is, what do you report? Is it just the accidents and the exposures, or are you really reporting near misses? Because if every laboratory could get people to actually report the near misses where something almost goes wrong, but you stop it before it does, those are the times that it's really valuable, because that's where you do have an opportunity to stop something where somebody realizes, oops, this almost happened. Well, we need to do some more training on this, or this is a situation that got me, kind of stuck here. And so if you can think about maybe reporting those near misses, and maybe some of you already are, or all of you are, but who do you report to, and how long do you have to file that report? Do you have to do it within so many hours? If you know where those forms are kept and what the forms look like, maybe you need emergency medical treatment right away. So where do you go for that? Do you just go down to the ER in your facility? Will the ER doctors know what you do in the lab? Do you need to take some kind of paperwork with you that explains, this is what I do in the lab, these are the organisms I might be exposed to, and these are kinds of symptoms or problems I might be having? So where do you get all that? And the important thing is, when we're talking about reporting incidents, is you always want to make sure, if you're a laboratory administrator, that you are never assigning any blame. You want people to feel really free to report incidents like this and exposures. They never want to feel like, oh my God, I made a mistake, I can't tell anybody, I'm going to get fired, or I'm going to get in big trouble. You want a very open policy in your laboratory where everybody is free and feels safe in reporting incidents. And then you can look at things and say, okay, what happened? Let's try and get to the bottom of this. What can we do better here? So let's learn from stories from a couple of past incidents I'm aware of. And they're kind of fictionalized or mixed up a little bit so that I'm not identifying any one laboratory, but you will get the points. So the phone rings, and what I often will hear is, oh, I think we have a possible BT agent. So when we hear that, and we're trying to do a better job, I've been the one asking these questions, but we actually have a team of people that are on our select agent team within our lab, and we're trying to get these questions so more people know what to ask up front if you call our customer service number instead of me or someone else. So we're going to ask about, what was the specimen source, and how old is the culture, and what media or plates is it growing on? What did the colonies look like? What's the gram stain result? What testing have you done so far? And did you do any of this work outside of the biosafety cabinet? So we're getting an idea of what you've done, where you've been working with it. What do you know about the patient history? And then the critical one that you're going to hear from me and hopefully from other people too is, did you contact the physician? If you're really thinking that this is a possible BT agent, contact that physician and tell them, this is what I'm getting. Is it possible, do these patient symptoms fit this organism? So you can get that going with the clinician right away and that physician. And then if it does, if we're assessing, this is a pretty high likelihood that this could be a Brucella or Francisella, which are the two main organisms that we see in Wisconsin in laboratories, and we seem to be getting more and more frequent calls with these. Do you have someone who's certified in packaging and shipping? Because you need somebody that's certified to package and ship a suspect category A specimen. And then when would we expect that specimen? How are you going to get it to us? Because we want to start thinking on our end about who we have to test on our side. Are we going to be prepared to do that? And then we'll ask for basic patient name, date of birth, and if you have any other questions for us. So just briefly in talking about packaging and shipping. If you truly believe and you are shipping something to us that you think is going to be a true Brucella or Francisella or Burkhold dairy or Yersinia or anything that you really think is probably going to be that agent, you have to categorize that package. And it's your responsibility as the person as that shipper categorizing it to think about the repercussions of what that is. And if you look at the regulations and just look at the classification chart, it will only say that you have to package something as a category A once it's a known BT agent. But if you're suspecting that that's truly what it is, then you have some kind of an obligation to package that as a suspect. And there's actual additional language in the regulations that talks about that, and they have information that says that that's what you should be calling it. You don't tell anybody, suspect anthrax or anything like that, but you do say it's a suspect category A organism isolate. So if you do that, there's a couple of things that are additional. There isn't a lot of difference between category B and category A as far as triple packaging. That's similar. They're both packaged similarly that way. But with the category A, there are some extra steps to keep it a little bit safer. You use the boxes that have that labeling on the outside that say it's been certified. They pass the drop box together. You actually have you have a different label, obviously, for category A versus category B. But you also have some information about emergency, who the emergency contact is if something happens with that box. And that emergency contact has to be available and answering that phone the whole time the package is in transport. It can't go to an answering service unless somebody at the answering service has all that information and can give it right away. It's not like you can have a page like we have with our emergency pager and we call you back. That doesn't work for them. It has to be a phone that's picked up that somebody can provide the information right away. You also have to have that shipper's declaration form that says what's in that package. And this is all just because if there would be, and heaven forbid that there ever would be an accident where a package like this breaks open, it's going to be a whole different cleanup for hazmat and how they're going to handle that. And you would want it to be that way because you wouldn't want, if your son's a person on, somebody that's on the cleanup and they're just doing normal cleanup and it ends up being something more hazardous, you want them to be appropriately in the proper PPE for a more hazardous organism that they're cleaning up. So think about that when you're deciding how to package something. And this is just another picture that just shows another picture of category A and how that is all put together. So you've gotten the specimen, you've notified us it's coming, we figured out when it's coming, and you've contacted not the Millennium Falcon, which I wish we could all, just fly it to you like something cool in Star Wars, but it doesn't work that way. So we have Purple Mountain Solutions, and they will pick it up for you. Obviously, if you're calling before noon, they pick it up that evening or late day in the afternoon, and it gets to us the next morning. Now, occasionally something comes in that is probably really urgent. Maybe you figure out on Thursday that in the afternoon, late in the afternoon, and it's too late to call them anymore for that same day pickup, you think, but you want it to get to us, and we are staffed on Friday to do that, but not necessarily Saturday. We would rather have that package getting to us on Friday so we can get it. So in occasional instances, we will work with you to get something with a more expedited delivery with Purple Mountain Solutions, but we have to request that of them, so you have to let us know, and we have to make that. You can't just say, well, I need this to get to the lab today. You have to do, this fast pickup. So work with us if that's the case ever. And so what happens when it comes to us is going to vary. It's dependent on several different things. That likelihood of an isolate being a true BT agent, the age, the pureness, the quality of the isolate when we get it, and how well it's growing. And again, is there an extra urgency for testing because there have been possible lab exposures in your laboratory? And there are also at least one test that we do not do at the Wisconsin State Lab of Hygiene if you are sending us botulinum toxin that actually goes to an appropriate national lab, which in our case is typically over to Minneapolis's lab. So that's something that would get sent on and forwarded somewhere else. So let's talk a little bit, okay, so that you can see some insight what's happening in our laboratory when we get these things. Our goal is, again, to give you the fastest answer possible and get this either ruled in or ruled out as a select agent. So we're going to always subculture whatever you send us, just because we may do more testing and we might want more of the bugs, so we're just going to subculture it. And then we often will repeat that rule-out testing you've already done. And there's a couple reasons we do that. We want to confirm the results that you got, or we may want to complete testing that you couldn't do in your laboratory. And then, again, just confirm that the correct isolate was submitted. Sometimes we do see different results in our laboratory, and we do a lot more of these than you do, so we will sometimes get a different result than you did. But we will kind of work through that flow chart too. If possible, then we're going to go directly and form an agent-specific PCR. If it really looks like our quick results and your results look like it's a bacillus, we're going to do that specific PCR for bacillus, or not for bacillus, I'm sorry, for Brucella. Saying the wrong one here. For Brucella. So the thing is, our PCR is going to call it a Brucella species. It's not going to give you which one it is, if it's a Suis melitensis. And I always forget the other one. Mortis. Okay. So we can't tell you that, but we can report a PCR, at least a result that it's a Brucella species. We will give you that. Some of the others are specific, right away. We can tell you that. But in most cases, that's the testing we're going to do. So we're working, and different people in different parts of the lab are working on different things, trying to get you this ID. We will maybe perform some other conventional testing in our laboratory too, depending on what it is and what we think it is. So just to be sure that you know that. And notifications are another thing. We're working through, as we develop our select agent team, we're working through some things now as far as, who's responsible for what and how we're doing these things. And unfortunately, because there isn't a set, this is how we always do it, and it varies, it's a little bit difficult. We're finding it's a little bit harder to figure this all out. But we will give you a positive result upon identification of a specific agent. So we're going to give you it. It could be a final, it could be a preliminary report. So as I said, you could get a positive report of a Brucella PCR, but that would be a prelim because we are still going to go ahead and work to try and speciate it then. So we're going to go and do other work that it will eventually give you a final result of which Brucella species it is. There is one exception. Bacillus species. All we're going to ever do with Bacillus species, if you're sending us something you can't rule out as anthrax, we're going to tell you it's either Bacillus anthracis or it's a Bacillus species that's not anthracis. And we aren't going to take it any further than that. We're not going to even try because there really aren't good tests out there to speciate that in our laboratory. So don't even ask us to try and speciate that. We just can't do it for you. Wish we could, but even the sequencing, it just isn't very good. So a final positive report, then again, you're going to receive a written report. You're going to get it either through the ELR, fax, or whatever standard method. And once you get that final ID from us, then there's a couple things that go into play right away. You in your laboratory have 24 hours to report any laboratory exposures to the Federal Select Agent Program. So because we will be reporting our ID to them as the final ID, that triggers them. And so they are looking for you to report if you have any exposures within that 24 hours. You have 7 days to complete. We complete something called FS, or the Federal Select Agent Program Form 4. So we start sections A and B of that, and then you will have to complete sections C and D. So we send that on to you to complete your parts of that. And that there are 7 days that you have to turn that in. And then you also have 7 days to gather and destroy all cultures, all original swabs or whatever you received for the source, the samples, anything where that organism is. You have 7 days to gather and destroy all those materials. Now, when we have a negative result, we will call with a negative result when we've ruled out all of the following select agents and toxins. So you're going to see the whole listing here of all of the different bioterrorism agents. The Burkholderia, Yersinia pestis, the Brucellas, Francisella, Bacillus anthracis, and then potentially, if you sent us something for Ebola or orthopox viruses or ricin toxin, you're going to get those negatives. The final negative will tell you that they've all been ruled out. And in a rare case, if it's something that we feel that we can identify by our MALDI, and keep in mind we haven't verified all organisms on MALDI, so we can't report on everything out. But if it's something we can report out, we will give you that ID. Then additionally, if we don't report that ID and you do want a final ID, and it's one of these other gram negatives, you can request that we do 16S sequencing. This is going to be a fee for service kind of a test. And it's not going to be done rapidly because we don't do it rapidly in the lab. It's going to take maybe 10 or 14 days before you get a result. So it may not be very useful for you for your patient care. It may be faster for you to just use whatever other methods you currently use to identify strange organisms. As I said before, we won't ever perform any of that on a bacillus species. So are there any questions about that? Because I know this kind of gets confusing. No, okay. All right, then just quickly, there are a couple of other handouts that were in your folder that were basically on CDC's website about suspected select agents and some flow charts. And these can be, I just threw them in there. You may find them useful or not, maybe to include in your policies or just use as a checklist for staff or something, but just all information that's there to help you with this. So once we've given you that ID and we know that you've got a select agent, now we're going to be asking about exposures. So did you have exposures in your lab? And what I used to always find out was that laboratories didn't think they have exposures. And then we started using a tool and asking all the questions. Well, did you do this? Did you do that? Did you do this, that, this, that? And where were you working? Were you working in a biosafety cabinet or were you working on an open bench? As we asked people to fill out those forms, they actually realized that they had exposures that they didn't really realize. And so we have a tool. This tool is actually a tool that there were a group of us on a committee through APHL that started working on developing this tool years ago now, and it's finally been released by APHL, and it is on their website. It's a great tool. I think especially for the exposure monitoring piece of it, there is the link at the bottom of the slide there that you can just link to that and look at it. I have a copy of it if anybody wants to look at it, but it's like it's a large document, so I wasn't going to print it out and put it in everybody's folders. What it breaks down is the exposure evaluation thing is very similar to what we're using, where it asks questions about what you were doing and where and who else was around you. And then for the treatment and monitoring, it actually breaks it down by a lot of different organisms. So there's a section in there for hantavirus, there's a section for Ebola, there's a section for this and that and everything, and it helps those of you in infection prevention and employee health and whoever else might be following up on that to kind of look at that. And it also gives you potentially a record for keeping track if you're giving prophylaxis, or are you monitoring temperatures, or whatever you're doing. So it will kind of help you work through that. So it is something I recommend highly that you go back and look at in your laboratory. This is another piece of the exposure monitoring guide. They broke out a separate piece that's a cute little poster. I actually have it hanging in my cubicle, but it does, you can see some of the different agents that they talk about. So it's got anthrax, blastomyces, psittacosis, botulism, C. difficile, acute fever, all sorts of different things. And it gives you some quick information about what would be the symptoms, what's the incubation period, and those kinds of things. This is actually the exposure assessment form that Rachel Close, who was the vet at the time, the state vet at the time, this was a precursor of this other tool that is completed now that we've been using in Wisconsin. And we just put it in a format that we can read it a little bit easier and look at it. So this is what we will actually be sending you, is this form to complete, and then you'll fill it out and return it to us. And if there are any exposures, we will be going over it with you and talking to you about it. So trying to help you through what might be the next steps for you too. And again, just a reminder of the forms you need to complete. Form 3 is only completed if there were exposures in your laboratory, and then you have that 24 hours to notify the Federal Select Agent Program. This has been problematic. I have had a lot of different labs have problems with this because sometimes they've tried to report, like when we give them that initial ID of just a Brucella species and they know they've had exposures, because I will start working with them then on exposures, and they'll report to the Federal Select Agent Program, but then they haven't had a final ID from us in the lab at all yet. So then they give the clinical lab grief about, well, we don't have any reports of a Brucella, because we just give them that final ID. So then the lab's confused. It's like, well, I thought I had to report this. So wait until you get the final ID, but then you have that 24 hours to report to them. If you don't report to them, on the other hand, other labs have gotten contacted, why didn't you report this to me within 24 hours? And so they do watch this. I don't know how they have all the personnel to do this, but they do watch, and they may be calling you saying, you didn't report this on time. So I'm just giving you that warning. And the Form 4, again, it's always completed when we identify an agent. We do the A and B sections, and you will be doing sections C and B. So we will work with you to get that form going. So this is another aspect. What does the Federal Select Agent Program expect laboratories to do with cultures growing a select agent? To A, dispose of the culture media in a biohazardous container and follow your facility's routine disposal of biohazardous waste. B, to package and ship all positive culture media to your LRN laboratory. C would be to spray your cultures with a disinfectant and then place in the biohazardous waste and follow your facility's routine disposal of biohazardous waste. D would be to use a validated method of autoclaving or overnight chemical disinfection with bleach to ensure you've killed all organisms and then dispose of as you would normal biohazardous waste. Or E, to call your biohazardous waste handler to see if they can autoclave and dispose of plates and other materials for you. So, what do you think is the correct answer? Okay. So the majority of you were able to tell that D is the answer that I am looking for. So they do expect that you are either going to use a validated method of autoclaving or overnight chemical disinfection with bleach, and that anything that leaves your laboratory is going to be killed before it leaves your laboratory. So to think about this, whenever you get a positive culture result, maybe you have a blood culture bottle, but that turns positive, you want to go back and start thinking about, well, were there other cultures that they might have sent later? Are there wounds? Were there second blood cultures that they sent? Make sure you're kind of looking and gathering all the materials on that patient that may indeed grow this kind of an organism. So gather them all, and then again, they have to be killed in your facility before transporting off-site for disposal. And I bring this up because I found out that somebody didn't have an autoclave, and so they mentioned it to people in their environmental services that handles, waste disposal, and they said, oh, we can just take that and send that off with, our waste hauler, and they'll take care of it. They'll dispose of it there. They didn't realize that they were in violation, that this, because it's a known Bt agent, now it is category A hazardous waste, and it can't go and leave your facility until it's killed. So, it happened. It's like they didn't know any better. So I'm telling you, you can't do that. You can't just have somebody haul it away for you, because you'd have to have it all packaged up as a category A, and you'd also have to get an approved transfer form at that point. So you'd have to have a federal select agent form 2 filled out and accepted by the select agent program to transfer it somewhere else. So you have to destroy it within the 7 days. Okay, so the best thing that we were able to do is get you this form. It's hot off the presses, and it's going to be again in this book that we're revising, but they actually posted it as a separate form just recently on the APHL website. This is a draft that came out like a day or two before they actually approved it, and I just needed to get it going. So just know it's a draft. You can go on their website and look for a different one, and I can put that in an upcoming message. But it talks about how to decontaminate waste, because labs are really confused on how do they do that safely. So if you're a non-registered clinical laboratory, that means you're not registered to possess by the bioterrorism types of agents, you're not going to keep them in your laboratory, then you have to kill these things. And inactivation has to be by autoclave or chemical decontamination. Now it says that it has to be a validated method, and so we were a little concerned about that, because it's like, well, are we going to expect clinical labs to validate that the bleach is really killing these? Nobody wants to do that kind of a study. So they came back and said, no, we're going to use this document. This is a best practices method, so you're going to be able to go ahead and do this, and they don't expect you to have to validate that as a clinical laboratory on site. So if you have plates and tubes and samples, what you want to do is just get a big container and fill it with 1 to 10 bleach solution. Do this in a biosafety cabinet, and then slowly put all of your media or whatever you're going to put in there, uncap it all so that that bleach solution gets into everything where the organism is growing, and let that sit overnight in a biosafety cabinet. Put a sign up so people don't go in there and move it around or take it out, because it just needs to sit there overnight. Then the next morning, you can safely dump that bleach out down the drain if that's allowed in your facility, if you can put bleach down the drain, and then get rid of the solid components in your normal biohazardous waste. So that handles that piece. If it's a blood culture bottle, and you don't have an autoclave, then what you're going to do is you're going to, again, take your bottle into the biosafety cabinet, and you're going to use straight bleach this time. You're not going to dilute it. You're going to put a piece of gauze, a disinfectant soap gauze, over the top of the bottle. You're going to put in at least 50 mLs of straight bleach into that bottle, and you're going to let it again sit in that biosafety cabinet overnight, and just let it sit there to decontaminate it. And then the next morning, it should be all killed, and you can take those bottles out and dispose of them as you normally would with your biohazardous waste. So, any questions about what you need to do to get rid of these bugs? Yeah?
I just have a question. And I guess what's the incident for labs of handling biosafety or hazardous chemicals like that? Organisms, sorry. I'm not a lab person.
Yeah, no, that's okay. We do not have a wet lab in Wisconsin that we actually have the ability to have labs work with these agents in their laboratories. We just actually really provide the information and then are there available as the resource for the questions, trying to provide, the information. I don't think it's anything that's too unusual for a laboratory to do. Does anybody think it's really odd, any of this that you're hearing? Pretty normal stuff you can handle, okay? So yeah, no exercises exactly, but just by the fact that we're providing the information, that's really what we are held to do, is providing the training and how to do things. So I wish we had a lab, but that's just not something that anybody has the kinds of dollars for these days. Okay, so we're done with that. Let's go ahead and move on to exposure monitoring and prophylaxis. Again, somebody who's ever on your team who this is assigned to, whether that's somebody in the lab, I think it falls to infection prevention or employee health to kind of do this piece of it. And it may be very personal depending. Some people may want to take prophylaxis right away. Others may not want to because, all of these antibiotics have side effects and for whatever reason you may or may not want to take something. And so that's something that you will be working with, doctors, employee health and stuff to decide. And then you do want to keep records, though, of who's, been exposed and how it was handled for them afterwards. Okay, so the next piece I want us to talk about for just a few minutes is, does your laboratory perform a root cause analysis after a laboratory exposure occurs? So A is yes, B is no, C is I don't know what management does, D is what is a root cause analysis, and E is both C and D. Okay, let's see. So many of you do. That's great. A couple don't really know what management does. And maybe some don't do anything. Go away. Okay, it doesn't like something I did. Yeah, it's turned itself off. That's what it did. Thank you. I figured that out just as you said that. Okay, so let's talk about root cause analysis. So if you think about root cause analysis, it's like having a weed growing in your yard. You know there's a problem. You've got an ugly weed. Something happened. And you can just do a real surface, kind of thing and get rid of it. Or if you don't want it to come back, that might not really get to the root cause, and you may still have another incident if you just deal with the surface issues. So you have to get down and dig out those weeds like you do with dandelions or something else in your yard that's a weed that you're trying to get rid of. So this is just one form. There's lots of different forms of root cause analysis you can do, but this is one where they suggest you ask why 5 times. So this is an example of, you're going to ask, why was there an exposure in our lab? Well, there was an aerosol created when they were spotting an isolate on an open bench for moldy. So why did they do that? Well, they were trying to get a really fast result for the physician, so they wanted to get that done, and they didn't really have any policy on doing a gram stain first or anything. So then you ask, well, why would that happen? And well, they didn't suspect it was a BT agent, it was a synovial fluid in a blood culture bottle. They weren't thinking about BT agents in a synovial fluid. And so then you keep digging down why. And well, there's no policy, again, to do a gram stain routinely before performing moldy. They didn't see that it was a small gram-negative, coccobacillus. And so then why, again, they missed clues that it was slow growing and they never looked at the patient history. So you can kind of dig down and look at what's happening for why people are doing certain things. And maybe, speed is emphasized more in the laboratory than safety. And if so, do you need to change that? Does something need to be adjusted a little bit here or there so that maybe you institute that when it's a slow growing bug, you have to do the gram stain before you do the MALDI on it. It's just so you get some kind of clue of whatever. We're not telling you how to do this or prescribing it, and we just want you to look at it and try and figure out what's going on in your laboratory. And one of the reasons that you're going to repeat a risk assessment, I said you look at that and repeat it periodically, is whenever there is an accident, if somebody gets a lab-acquired infection, if there's a theft or a security violation with an agent, that you are going to do a repeat risk assessment and look at what happened. So you've got some gaps you potentially identified from your root cause analysis, and you're repeating your risk assessment. And this is the classic repeat risk assessment cycle that you see where you've identified new hazards and now you have to evaluate how likely is this going to happen again, what are the consequences again, and so what other further mitigation steps can we put in place. So in this case where people are working really fast with speed as a priority, there's a high risk this is going to continue to happen because, you're still got the same patient care issues that are out there. So again, what do you put into place to further mitigate the risk? Well, again, maybe you can spot your MALDI plates in a biosafety cabinet. You can institute that gram stain at the same time and actually read the gram stain before you spot the MALDI. And whatever it is, provide further training to people on some of these stopping steps so that they remind people of what to look for. Going to implement those new controls and then evaluate the effectiveness of your new risk assessment. So this puts us at the end pretty much. Really the goal here, and my goal has always been since I took this role at the state laboratory, to protect the most valuable piece of all of our laboratories, and that's all of you, all the people who work in the laboratories. Without you, our laboratories, they're just laboratories. Nothing gets done. And so we really want to keep all of you safe, and sometimes that means extra steps that maybe slow down the work, and yet if there's a true trade-off in the value of keeping you safe, then you need to do those kinds of things. So here's our laboratory being safe during COVID, and then all the Jedi warriors with their little sabers. I don't have lightsabers to give to all of you to keep you safe, but I wish I did. So just a couple of key points, and these are the key points that are on the paper for me for this presentation. That your biosafety plan is your strategic plan in the lab, and biothreat agent biosafety awareness training should be provided initially to new hires and then thereafter as part of your yearly refresher training. And it is critical in preventing laboratory exposures or actually acquired infections or associated infections, I guess they say now. You want to identify that exposure response team, your occupational health team, however you call them, in advance. So if you don't know who a team is now, go back into your laboratories and identify those members, get all that contact information available, and put it in a place that all the laboratory knows where that is. You can always call us for help. We are your LRN reference laboratory, and we are happy to help. That's why we have our 24/7 pager number that you can call and contact us. We also have a great customer service team that is available during office hours, so those numbers are there. And we will assist you in identifying any suspect biothreat agents as quickly as possible and work with you to get that process completed. And also to help you assess your exposures and any guidance with destruction of the organisms. And then just remember to perform that root cause analysis afterwards. And you're, looking at improving things and preventing these same things from happening over and over in our laboratories. So that was a lot. At least it's a little bit before lunch. Now you can go and think about it and maybe talk about it a little bit more at lunch if there are questions that come up. But I do know that one of the stories that labs have is when they didn't have contact information available, somebody thought they'd been exposed to a Neisseria meningitidis on the open bench. They set something up, and so they just went down to the ER after their shift. There was no one around, no supervisor on staff or anything. And unfortunately, what they got in the ER was, well, who do you know that has Neisseria meningitis? They're thinking of people exposure in the normal sense of a patient coming into the ER. And they're like, well, were you at a party? Were you sharing beverages? Were you kissing the person? You know, close contact? And it's like, no, I work in the lab. And the clinician was really kind of clueless as far as, okay, our exposures are different. Our exposures are from working with the organisms, and we can breathe them in and get them other ways. Or we can have a needle stick or whatever. Maybe the needle stick would have hit them more, but this is the kind of reason that you want staff to know ahead. Who did they contact? What did they do on an off shift? They didn't want to call the supervisor. It was a weekend, and they were reluctant to do that. So make it really evident in your laboratories what your policies are, and it'll help the staff a lot feel more comfortable in the fact that you care for them. So, all right, one more time. Questions? Over there.
So if you have a potential agent in blood cultures, I would assume it would be safe to say this patient also has other tubes of blood floating around in the lab. Should those also be destroyed?
Yeah, that's a tough one. There's not a lot of clear guidance, but I think you should have a healthy suspicion of other samples that may have gone to hematology, to chemistry. When you're going to collect samples from a known positive, collecting them from everywhere and destroying them is really what you want to do. And as you're thinking about potential exposures, I find, unfortunately, a lot of core labs don't necessarily have face shields or plexiglass between them and the samples that they're working with. So those are other potential lab exposures when you have known positives. However, I do want to give people a heads up. By the time you get a result back from us, a confirmed positive, most of these have already been destroyed. I don't know how long you hold on to samples in your lab, but if you're high volume, there's not a lot of space to hold on to too much. So thinking about that, even when you have a suspect, start thinking about where are those samples? Can you round them up now before they're gone?
Thank you.
Great question, and something I meant to bring up, so I'm glad you asked. Anyone else? All right. If you've listened to the entire morning session of our 2024 regional meetings, and you're looking to get PACE credit for listening to these presentations, you're going to need to go to the ASCLS CE Organizer website. And once you get to their website, you're going to click on the tab for other PACE providers and search through the listing of the other providers to find the provider for the Wisconsin State Laboratory of Hygiene. Then you'll click on the View Events, and you will see the listing for the morning sessions of the WCLN regional meeting. When you find the morning session and click on it, the session code number that you're going to need is 9185. Again, that session code number is 9185.